Hello, this is the final part of chapter 30, which is part 3. Now, we're going to continue talking about seedy plants, and we're going to talk about angiosperms or the flowering plants. The reproductive adaptations of angiosperms include flowers and fruits. They are the most widespread and diverse of all plants. Extant seedy plants form two sister clades, clade gymnosperm and clade angiosperms. Clade is equivalent to monophyletic group. Monophyletic group or clade should have common ancestor. This is common ancestor for these two groups. Recall that gymnosperms have naked seeds exposed on sporophylls that usually form cones. Angiosperms or angiosperm seeds are enclosed in chambers that mature into fruits. All angiosperms or the flowering plants are classified in a single phylum called Anthophyta. Anthus means flower, phyta. The life cycle of an angiosperm is a highly refined version of the alternation of generations common to all plants. Plant life cycles are characterized by alternation of generations. The first generation is the diploid sporophyte, which is dominant in vascular plants, alternate with the haploid gametophyte generation, which is highly reduced in these vascular plants. Sporophytes produces haploid spores by meiosis, while gametophytes produce gametes by mitosis. Gametes, sperm in the male gamete, and egg, as you know, the female gamete. In angiosperms, the sporophyte again is the dominant generation. They are larger, more conspicuous, and longer lived than gametophytes. The angiosperm life cycle is characterized by 3 Fs: flowers, fruits, and double fertilization. Flower structure and function. Flowers are specialized reproductive shoots of the angiosperm sporophyte. They attach to a part of the stem called the receptacle. Many species are pollinated by insects or animals, while some species are wind pollinated. Sometimes a human can pollinate some plants in experimental, for example, genetics. Flowers consist of 4 types of modified leaves called floral organs. Sepals, sterile modified leaves, which enclose the flower. Petals, sterile modified leaves, which are often brightly colored to attract pollinators. Wind-pollinated flowers generally lack brightly colored parts. Stamens and carpels are sporophylls, leaves specialized for reproduction. Stamens, the main reproductive organs. A stamen or a microsporophyll consists of a filament and an anther. Microspores, which are produced in the anthers, develop into pollen grains containing the male gametophyte. Carpels, the female reproductive organs. A carpel is a megasporophyll, has a long style with a sticky stigma on the top that captures pollen grain during pollination. At the base of the style is an ovary containing one or more ovules. The ovary contains the female gametophytes within the ovule or ovules. Fertilized ovules develop into seeds. The term pistil can be used to refer to a carpel or two or more fused carpels. Again, this is more clear. This is the female reproductive organ, the carpel or the pistil, stigma where pollen grain lands, style, and ovary. Inside the ovary we have many ovules. The male reproductive structure is the stamen, consists of anther. In anther chambers there that later on the male gametophyte are developed and release pollen grains. And this is the filament. Anther and filament forms the stamen. The following video shows time-lapse flower blooming. You can observe this. Flowers are variable in shape, size, color, and odor. For example, some flowers have radial symmetry, while others have bilateral symmetry. In radial symmetry, any imaginary axis or any imaginary line through the central axis divides the flower into two equal parts. Here we have more than two equal parts. In bilateral symmetry, the flower can only be divided into two equal parts by a single imaginary line. Fruits. A fruit is formed when the ovary wall thickens and matures. Fruits protect seeds and aid in their dispersal. Mature fruits can be either fleshy or dry. Examples on fleshy fruits: tomato, a fleshy fruit with soft outer and inner layers of pericarp, the fruit wall. Ruby grapefruit, a fleshy fruit with a firm outer layer and soft inner layer of pericarp. Nectarine, a fleshy fruit with a soft layer and hard inner layer inside here of pericarp. Examples on dry fruits: milkweed, a dry fruit that splits open at maturity, as you see here. Hazelnut, a dry fruit that remains closed at maturity. Various fruit adaptations help disperse seeds. Seeds can be carried by wind, water, or animals, sometimes humans, to new locations to ensure that seeds germinate away from the competitive influence of the parent plant. Dispersal by wind. Here we have few examples in this. Giant seed of the tropical Asian climbing gourd, you see here. Again on the dandelion, seeds actually one-seeded fruit. Also we have a winged fruit of a maple. This wings enables the fruit to be carried by wind, make it easier. Some plants have mechanisms that disperse seeds by explosive action, like in this. We have another example, a plant called Equilibrium elaterium, a cucumber, it's a type of cucumber. And when you touch this type of cucumber, it explodes and release the seeds. Dispersal by water, the coconut seed embryo, endosperm and endocarp inside this. Dispersal by animals. Seeds within berries and other edible fruits are often dispersed in animal feces. Here we have seeds dispersed in black bear feces. Squirrel in this picture hoarding seeds or fruits underground. Ant here carrying seeds with attached food body. Again, the barbs here of cockleburrs facilitate seeds dispersal by allowing the fruit to be attached to animals. The following animation shows fruit development.
When seeds are mature enough, the ovaries change their function and develop into seed-dispersing structures called fruits. There are many patterns of fruit development, depending on the number of carpels, ovaries, and flowers that form one fruit. Fruits can also be divided into the categories of dry fruits and fleshy fruits. In dry fruits, the ovary dries out at maturity. It may develop hooks that catch animal fur, as in this example. Or the ovary and other flower parts may form a wing or parasol or hairs to catch the wind. Sometimes the dry ovary builds tensions that, when released, will shoot the seeds into the air. In fleshy fruits such as tomatoes and peaches, the outer tissues of the ovary thicken and become soft, sweet, and brightly colored. Fleshy fruits invite animal dispersal. The seeds are ingested and, if they escape digestion, they are excreted with fertilizer, hopefully in a choice location.
The angiosperm life cycle. An overview of the life cycle. The flower of the sporophyte is composed of both male and female structures. The angiosperm life cycle includes the following, and we're going to talk about each step in the following or in this lecture. First, gametophyte development. Second, sperm delivery by pollen tubes. Third, double fertilization, a characteristic feature of angiosperms. And finally, we're going to talk about seed development. This figure shows the angiosperm life cycle. We start now with gametophyte development. We're going to talk about male gametophyte development and female gametophyte development. As we mentioned before, angiosperm gametophytes are microscopic, highly reduced, and their development is obscured by protective tissues. Now, the development of female gametophytes, which is the embryo sacs. The female gametophyte or embryo sac develops within an ovule, this ovule, contained within the ovary, an ovary at the base of the stigma. Here the stigma, the style and ovary. In this ovary, we have the ovules, and the gametophyte, female gametophyte, will develop within these ovules. Within an ovule, two integuments surrounded the megasporangium, except at a gap called the micropyle. One cell in the megasporangium, the megasporocyte or the megaspore mother cell, undergoes meiosis, producing 4 megaspores. One survive and give rise to female gametophytes. To female gametophyte, the other 3 will disintegrate. The megaspore divides, haploid megaspore divides by mitosis without cytokinesis, producing one large cell with 8 haploid nuclei. The cell is partitioned into a multicellular female gametophyte called the embryo sac. This one. Female gametophyte or the embryo sac consists of 8 nuclei and 7 cells. The central cell with 2 nuclei. We have at the top 3 antipodal cells. Down here, we have an egg surrounded by 2 synergids, 2 cells. Female gametophyte do not produce an archegonium, are located within an ovule, consist of only few cells, an embryo sac with 8 haploid nuclei in 7 cells, a large central cell with 2 haploid nuclei, and one egg cell here. This is again the embryo sac. Remember that this is the embryo sac is the female gametophyte, highly reduced into 7 cells and 8 nuclei. The development of male gametophytes in pollen grains. Male gametophytes are contained within pollen grains produced by microsporangia of anthers. Here's the anther. Okay, this one. The anther is formed from 4 chambers here. Not very clear, not found very clear, not seen very clearly, but in this area. Pollen develops from microspores within the microsporangia or pollen sacs of anthers. Each microspore undergoes mitosis. Microspores are haploid cells. Divide by mitosis to produce 2 cells. The generative cell later on divides forming 2 sperm cells and the tube cells that produce pollen tube during pollination. Therefore, notice that here is a mature male gametophyte consists of 2 cells only, highly reduced. A pollen grain consists of this, as you see here, this is the pollen grain or the male gametophyte, the mature one. Where here, spore wall surrounding these 2 cells, the generative cell and the tube cell over here, seen here. Okay. فإذا الجاميتوفايت found where inside this pollen grain. Now we're going to talk about pollination. Sperm delivery by pollen tubes. Most flowers have mechanisms to ensure cross-pollination between flowers from different plants of the same species. This increases the variations, which is very important source of genetic variations. A pollen grain that has landed on stigma germinates, and the pollen tube here of the male gametophyte grows down the ovary and discharges 2 sperm cells near the embryo sac, the female gametophyte. And this sperm will be delivered into each ovule. The ovule is entered through pore, as we have mentioned before, called micropyle. Over here, this is the micropyle, and again the pollen tube entered this micropyle and release the 2 sperm cells. Double fertilization occurs when the pollen tube discharges 2 sperm cells into the female gametophyte within an ovule. Fertilization is the fusion of gametes, male and female gametes, occurs after the 2 sperm reach the female gametophytes. This is double fertilization. This is the nucleus, the central cell, 2 nuclei, with one sperm cell forming the 3N endosperm. And here down, one sperm will fertilize the egg, forming the zygote, 2N zygote. The zygote will divide and form the future embryo inside this ovule. Later on, this ovule will become the seed. Look again. As we have mentioned earlier, this is the pollen tube releasing 2 sperm cells through the micropyle into the female gametophyte. One sperm fertilizes the egg, forming the zygote, the future embryo, or the future sporophyte. The other sperm combines with the 2 polar nuclei in the central cell of the female gametophyte, giving rise to the triploid food-storing endosperm, the nutritive tissue. The double fertilization ensures that endosperm only develops in ovules containing fertilized eggs. Therefore, the triploid endosperm nourishes the developing embryo. Within a seed, the embryo consists of embryonic root, and 1 or 2 seed leaves called cotyledons. This is the embryo. This is the embryonic root. This is the 2 cotyledons. Then the seed consists of this embryo, future sporophyte diploid, endosperm, nutritive tissue 3N, and the seed coat derived from the integument 2N. Of course, the integument is part of the sporophyte tissue. Sporophyte tissue is 2N. This animation shows plant fertilization.
The function of flowering is to produce seeds and disperse them to locations where the young plants are likely to survive. At each step, there is an emphasis on protecting the offspring. This activity reviews some of the most important steps. Events in seed formation begin with pollination, in which wind or animals bring pollen to the flower. The growth of pollen tubes caused the flower to discard its colorful parts, petals and stamen. After pollen arrives, the bright petals are a hazard. They attract animals that might eat the delicate young fruit. We've enlarged the ovary and sliced it open. An ovary contains 1 or more ovules that will become seeds if they are fertilized. One ovule is shown here, with the front removed to reveal its parts. The ovule is attached to the ovary wall by a stalk that provides nutrients. Starting from a pollen grain at the stigma, which is not shown, a pollen tube grows down through the carpel to the ovule. It carries 2 sperm that enter the ovule. One sperm fuses with the egg cell to make a zygote, which is the first cell of the embryo. The other sperm fuses with the 2 nuclei in the central cell, making a triploid nucleus. The triploid cell will grow and divide to make a food-storing tissue, the endosperm.
Seed development. After double fertilization, each ovule develops into a seed. The ovary develops into a fruit enclosing the seeds. When a seed germinates, the embryo develops into a new sporophyte. Here, the embryo inside this seed, embryo 2N, endosperm nutritive tissue 3N, triploid tissue, seed coat 2N derived from the integuments of the sporophyte tissue, of course 2N. The seed will germinate under optimal conditions, when conditions are suitable for this seed to germinate, giving rise to small young seedling, and the seedling later on matures into mature diploid sporophyte, the plant we see. This figure outlines the life cycle of flowering plants. To summarize this life cycle, Pollen from the anther land on sticky stigma at the carpel's tip. Most flowers do not self-pollinate, but have mechanisms to ensure cross-pollination, source of variation, which is good. The pollen grain germinates on the stigma by growing a pollen tube down the style of the carpel. When it reaches the ovary, the pollen tube grows through its micropyle and discharges two sperm cells into the embryo sac, which is the female gametophyte. Double fertilization occurs as one sperm nucleus unites with the egg to form a diploid zygote. This is the first cell of the embryo. The other sperm cells, or the sperm nucleus, fuses with the two nuclei in the embryo sac's central cell to form triploid 3N endosperm tissue. After double fertilization, the ovule matures into seed or into a seed. The seed consists of embryo. The zygote develops into an embryo with a rudimentary or embryonic root and one cotyledon or two cotyledons in dicots or seed leaves. We refer to these cotyledons as seed leaves. In the sperm, the triploid nucleus in the embryo sac divides repeatedly, forming triploid endosperm, rich in starch and other food reserves. Seed coat is derived from the integuments, outer layers of the ovules, and protect the seeds. Seeds will germinate when proper conditions or optimal conditions for them to give rise to the mature sporophytic plant. Here is shown the flower of this plant. The following animation shows seed development.
This ovule has been fertilized and will become a seed. Each of its cells contains one or more sets of chromosomes, but the number of sets of chromosomes differs among the parts of the ovule. Double fertilization converts the large central cell from diploid to triploid, while the egg becomes a diploid zygote. The surrounding integuments remain diploid as they were before fertilization. The embryo is a young plant that grows from the zygote, while the large central cell grows into food-storing endosperm. All this is enclosed within a protective seed coat that grows from the integuments around the ovule.
Taking food through its stalk, the developing seed expands its endosperm and embryo. When the embryo is mature and dormant, the integuments develop into a protective seed coat and detach from the ovary wall.
The main axis of the embryo has a root apex and a short stem that may have a few tiny leaves, not shown here, near the shoot apex. The cotyledons are part of the embryo. They're the first leaves. Monocots have one cotyledon. Eudicots have two.
The following video shows time-lapse of the life cycle of a flowering plant. Angiosperm diversity. Angiosperms comprise more than 250,000 living species. Previously, angiosperms were divided into two main groups based on number of cotyledons or seed leaves in the embryo, flower, and leaf structures, etc. Those features were used also to define the two groups until 1990. One group is the monocot with one cotyledon. The other group is the dicots with two cotyledons. Recent DNA studies, however, indicate that the species traditionally called dicots are paraphyletic. So systematists categorized the vast majority of species, once categorized as dicots, to form a large clade known as eudikots. Therefore, the clade eudikots, true dicots, includes most dicots. The clade, remember that clades equivalent to monophyletic, that's to say those organisms share the same ancestor or one common ancestor. Figure 16 compares the main characteristics of monocots and eudikots. The seed. Embryo with one cotyledon, while eudikots, the seed with two cotyledons. Leaf venation. In case of monocots, most leaf veins are usually parallel, therefore parallel venation. In dicots, venation is net-like or reticulate. The stems. The vascular tissue are scattered in the stems, while in the eudikots, the vascular tissue are usually arranged in rings or in ring. Now, monocot roots. Root system usually fibrous, that's to say no main root as you see here, while in eudikots we have main root called the tab root. Pollen grains. Pollen grain in monocots has one opening, while pollen grain of eudikot has three openings. Flowers. The petals of the monocots are usually multiple of three, while that in dicots are usually multiples of 4 or 5. The rest of the former dicots form several small lineages. Basal angiosperms include the flowering plants belonging to the oldest lineages. About 100 species composing 3 small lineages constitute the basal angiosperms. These include Amprolla trichopoda, water lilies, and star anise. Magnolids share some traits with basal angiosperms but evolved later. Magnolids consist of about 8,000 species, including both woody plants and herbaceous plants. Magnolids are more closely related to monocots and eudikots than to basal angiosperms. Example, here we have the magnolia. Monocots. About 70,000 species are monocots. The largest groups are the orchids, grasses like barley, wheat, rice, and so on, and palms over here. Eudikots. More than 2/3 of angiosperms are eudikots, about 170,000 species. Eudikots include the large legume family and the economically important rose family. This figure shows angiosperm phylogeny. The tree represents a current hypothesis of angiosperm evolutionary relationships based on morphological and molecular evidence, DNA analysis. Angiosperms originated about 140 million years ago. The dotted line here shown indicates the uncertain position of a possible sister group of extant angiosperms. Therefore, angiosperms, we have, as we have mentioned before, one phylum Anthophyta. Anthophyta, we have two clades, eudicots and monocots. Those have common ancestor. The rest of the angiosperms, a group called magnolids, and three basal angiosperms, Amborilla, Trichopoda, water lilies, and star anise, and the relatives, all forming, as we have mentioned, the basal angiosperms. Those are the remnant بقايا الدايكوتس. The dicots, they group the largest number into eudicots, and the rest are the Amborilla, water lilies, star anise, and relatives plus magnolids. Summary of key points of angiosperms or flowering plants. The flowering plant life cycle alternates between a conspicuous or dominant diploid sporophyte and a reduced haploid gametophyte, as in all seeded plants or as in all vascular plants. The angiosperm life cycle is characterized by three F's: flowers, fruits, and double fertilization. Flowers are involved in sexual reproduction. Female gametophytes are produced in the ovary. Male gametophytes are produced in the anther. Pollination is the first step toward fertilization. Many species are pollinated by insects, animals, and wind. Fertilization is followed by seed and fruit development. A unique double fertilization process occurs in the flowering plants. The mature seed contains an embryonic plant and storage materials. Fruits are usually mature, ripened ovaries. Seed dispersal is highly varied by wind, water, animals, or humans. Genetic diversity produced by sexual reproduction. All angiosperms are classified in a single phylum, phylum Anthophyta. Several groups of basal angiosperms have been identified: water lily, Amborilla trichopoda, and star anise. Other major clades of angiosperms include magnolids, monocots, and eudicots. Thank you for listening and good luck.